Thank you for the introduction. First of all, I would like to thank the organizers for inviting me to this exciting conference. Today I'm going to share some of our recent work about bacterial active solids and its emergent mechanical behavior. Our interest in this topic arose from our study of large-scale force generation in bacterial colonies or bacterial communities. As we know, large-scale generation and regulation of mechanical forces are crucial to the development and physiological functions of multicellular organisms. For instance, the cardiac tissue beating and the contraction of smooth muscle cells, muscle tissues, they pump liquid fluid throughout the body circulation system. And coordinated beating of cilia can generate macroscopic flows that help with mucosal clearance and waste disposal in the brain. But we are, instead of multicellular organisms like animals, we are interested in large-scale force generation in bacteria. Why are we interested in that? First of all, any form of large-scale force generation in a bacterial community can have impact on the material transport or dispersal of the colony itself. Also, the so-called engineered living materials has been considered as having potentials in biotherapeutics. And bacteria is being used as a premier building blocks for such kind of engineered living materials. So far, the study of the functions of these living materials is primarily focused on the biochemistry side, like the drug release, chemical effector release, or serving as biosensors for the environment. But if we want to engineer or design mechanical functionalities of such living materials, we need to understand how large-scale forces can be generated in such materials. But for bacteria, such simple creatures, it's very difficult for them to generate large-scale forces. However, they have invented different ways to do so cleverly. Here I give you several examples. For instance, a biofilm can make use of surface friction to regulate their morphology patterning, like the formation of these wrinkles. Okay. Underneath these wrinkles, there can be directed fluid flows that transport liquid from outside the colony to the center. And this is driven passively by the evaporative flux inhomogeneity on the surface. More recently, my lab discovered a form of active interfacial mechanics that can help centimeter-scale bacterial colonies to drive high-speed fluid flow. In this case, bacteria produce biosurfactants, and this biosurfactant gradient can trigger shear bending in an initially homogeneous colony, and that these shear bands develop into these fluid channels, appear as valleys in this photograph. So these channels, they are pretty much like canal structures built by bacteria, in which the fluid flow as high as several hundred microns per second is being driven from the center to the edge. All bacteria that can make use of the global mechanical environment to rectify their microscopic force generating processes. For instance, bacteria can form this type of veil structures. I'm not sure if you can see it clearly. So this thin sheet of thin stuff, it's called a veil. So motile cells attached to these veils can generate macroscopic flows. Similarly, at the very edge of a developing colony consisting of flagellated bacteria, the cells can self-organize at the edge of the three-phase boundary. Three-phase means liquid, gas, and solid interface. So this interface helps rectify individual cells' orientation to make them well-ordered that can generate directed flow around the colony to help with material transport. These forms of large-scale forces in the bacterial world, they all depend on the external global mechanical environment. And if we want to have bacterial-based living materials with mechanical functionality, programmable mechanical functionalities, that would be not ideal because it reduces the flexibility or controllability. On the other hand, if we look back at the multicellular organisms, like animals, a general strategy is to solve this problem is to couple microscopic mechanical processes and to, in order to have large-scale force generation and regulation. For instance, the coupling can be electrochemical coupling or mechanical coupling. Okay. We know bacteria have different ways to generate microscopic forces with their motility mechanisms, and whether this capability can be turned into some form of large-scale emergent mechanical behavior. That's the question that we would like to address in the following of the talk. So here I'm going to showcase two examples how bacteria can make use of their motilities to do the job. This includes one consisting of coupling of flagellar motility, and the other is the coupling of the so-called type 4 pillar motility. And the first one, flagellar motility, is quite well known to many of the audience. So flagellar are long, thin, helical-shaped filaments appended on cell surface. When they rotate in the fluid, they can generate self-propulsive motion. Much of our knowledge on the biophysics of flagellar motility was made through the work in Howard Bird's lab, where I did my postdoc about 10 years ago. Sadly, Howard passed away a few years ago. If we have flagellated bacteria embedded in an elastic matrix, this elastic matrix can be self-produced by the cells, such as when they form biofilms. Then it makes an ideal material to investigate the continuum mechanical behavior of the so-called bacterial active solids. Okay. Because now the cells are embedded in an elastic matrix, and their relative position is fixed. In such a bacterial active solid, my former student and postdoc, now an assistant professor at Zhejiang University, Haoran Xu, found that the mass elements in this material can be set into oscillatory motion. And if the confinement is isotropic, okay, like a disk, then these individual mass elements' oscillatory motion can appear in two global motion modes. One is the global translational motion, as shown here, where every mass element undergoes a circular trace. And the other is the global oscillatory motion, where the entire disk rotates with the center being fixed. So now every mass element traces a linear trajectory. Here a movie of the translational mode, where you see this translational motion with the center tracing a circular trajectory. And the other is the global rotational mode. Now the center is fixed. Okay. Here, every dot indicates one bacterium. Okay. Only a tiny fraction, maybe less than 100, less than 1%, is labeled. So you can imagine how dense this system is. Okay. This type of global motion modes can be produced also in macroscopic robotic analogs, like what we have seen in the morning. In addition, if the system is under anisotropic confinement, there can be standing waves. So if we look at the velocity field and decompose the velocity into two Cartesian coordinates, here is the transverse component, here is the parallel component, we can see that it's the standing wave is a transverse standing wave. So such standing wave can be better seen by looking at the space-time correlation, where we can see segregated domains with high correlation for the transverse component. All right. More interestingly, that when we tune the activity of individual cells, there can be a transition between the two modes. For instance, at the low, so here if we follow time, the time axis, we keep continuously decreasing the motility. So initially, at high motility, high activity, the system displays the translational mode. And at certain threshold of activity, it suddenly transits to the translational mode, the rotational mode. Accompanying this mode transition, there's twofold frequency jump. Okay. The translational mode has a higher frequency. And within each mode, the oscillation frequency is quasi-linearly correlated with the activity. Then we build a computational model for this active solid, bacterial active solid, consisting of flagellated bacteria. So in this model, the mass elements are denoted as active particles, and these active particles are connected by elastic springs. Okay. So each particle is described by its position and its polarity, orientation. And every particle experiences three types of elastic forces. One from the bulk, from the elastic springs connecting the particles, and the other from the interaction with the substrate, and the third coming from the interaction with the boundary. Okay, so we work with Ray Zhang at Hong Kong UST and his student. They help us design this free energy Landau type free energy functional that can account for the dynamics within each mass element that describes the alignment of individual force generators, okay, individual cells. So the polarity of individual cells by this description will spontaneously align with the global moving direction of the mass element. Right. With this model, we can reproduce the translation mode at high activities, or the rotational, global rotational mode at smaller activities, and also the transverse wave. And during the mode transition, so the model can also reproduce this frequency jump. Okay. And by scanning the parameters, model parameters related to particle activity and the elasticity, we can have a simple phase map that looks like this. So basically, at high activity and low elasticity, the system displays translational mode, and otherwise it displays rotational mode. And there's a sharp boundary in the oscillation frequency at the boundary of these two modes. Okay, so it's always roughly so the ratio of these two modes oscillation frequency is almost the same along this boundary. If we tune the parameters in the right way, then it's always roughly 2. Okay. Right. So the model predicts that the system's behavior depends on elasticity. This can be verified in experiments as well. So we use temperature as a way to tune the elasticity of the polymer networks. So temperature is So as temperature increases, elasticity also increases. And then we see this transition from the translational mode to the rotational mode. All right. So this is the story about how coupling flagellar motility through an elastic matrix can help us get some interesting emergent mechanical behavior. For the second part of the story, so we examine this type of motility called type 4 pillar motility. So different from flagellar, which are helical filaments, type 4 pillars are linear filaments. They are straight. So this is an atomic force micrograph of these motility organelles. So they are roughly several micron in length, comparable to flagellar. But the working mechanism is very different. Flagellar motors rotate, so there's a complex motor, rotary motor at the base of every flagellar filament. The rotation is driven by protomotive force. Okay. But this pillar motor, it's a linear motor. It expands and retracts in a cyclic manner. And the power source is ATP. Okay. The motor components include several proteins, several enzymes. These enzymes that consume ATP to generate the forces. Pillar motor has been recognized as the most powerful molecular motor known to date, because there's a study done 2 decades ago by Mayer and Michael Sheets. They found that the retraction force, the force generated by this motor, can be up to 100 piconewton. Compared by contrast, a rotating flagellar can generate a force up to 1 piconewton, 100 times difference. So it works by, as I said, it works by cyclically expanding and retracting. So the pillars, when it's extended, it acts like a grappling hook. It attaches to the body of other cells or to the substrate, and then pull along this fiber. So this way, the cell can generate movement. Although the force is quite powerful, but they operate this type of motility mostly on substrates near a surface. Okay. On surface, they have to overcome a very large friction. So the resultant movement is very slow. It's only several micron per minute. That's incomparable to the swimming speed of bacteria in liquid. All right. So here is the background about pillar motility. And when we study a dense collection, a dense film of bacteria consisting of these pillar-driven cells, we found a spiral wave. So this study was done by my former PhD student, Liu Siqi, who's now a postdoc at UC San Diego. So what we found is that under phase contrast microscope, we see periodic variation of light intensity of every mass element in this film, and this variation in space is organized as a spiral. So here's a video showing how this look like. Notice that the scale bar is 500 micron. So a single spiral wave can propagate at a speed about 2 millimeter per minute, and it can extend to several millimeters in distance. Right. So the period is roughly 6 minutes. Okay. And this spiral wave emerges through a self-organization process. So this video shows the developmental phases. So initially we see very irregular wave fronts appearing at random positions. And gradually these wave fronts interact with each other, they coalesce, and so some of them annihilate, and some of them strengthen with each other and becoming a single stable spiral. Okay. Eventually the system will leave only a few stable spiral waves, and these spirals at the steady state are very stable in the sense that the core of these spirals are almost stationary. This is in contrast to the physical...
Question?
Yes?
Sorry, I just missed what you measured actually, to measure the spirals. You said it's the argument of, the phase of what exactly?
So here the phase is defined by looking at the light intensity variation in space. So we know every mass element, it oscillates in its light intensity. So we denote it as an oscillator, a phase oscillator. And we can measure the phase distribution in space. Then once having this phase map, which is shown like this, then we can use this order parameter to define, to locate those spiral cores. Yeah, that's how we get this space-time trajectory of these spiral cores.
The question is, what is the fluorescent intensity? Is this some unit expression of the bacteria or?
So the question is about the fluorescent intensity. So here we do not have fluorescent intensity yet. So here this color map shows the phase of each. Oh, it's the wave front. The wave front are traced by looking at the phase contrast microscopy images.
So it's density.
Yeah. Yes, indeed, it means density variation. Yeah, we can. So we have done some measurement with fluorescent label cells, and to show that this is indeed corresponds to the cell density variation.
So I have a related question. Are you getting both chiralities in these spiral waves, or are you predominantly one chirality?
Yeah, that's a very good point. So here we get both types of chiralities in the same system. So the spirals can be either clockwise or counterclockwise. Okay, any further questions before I move on? All right, so this, as I just mentioned, this pattern has very stable spiral cores, and that's in stark contrast to the meandering motion of spiral cores in some non-equilibrium chemical reaction waves, like DZ reaction waves in excitable media. Now, we looked at the microscopic mechanism of this spiral wave. As I just mentioned, we label the bacterium with the fluorescence, and what we see is that as the wave propagates through space, there's a density variation associated with the wave front. Meanwhile, every mass element moves back and forth. The magnitude is rather small. It's not easy to see by naked eyes, but if we measure the trajectory and get the displacement, we can see that every mass element moves back and forth along with this wave. By treating, so that means it's a longitudinal density wave. By treating this quasi-2D bacterial film driven by Pillar's motility as an elastic medium, we can relate the tension force generated by the bacteria, which is due to the pooling. Okay, so Pillar's pooling generates tension. We can relate tension and the displacement of the film, and by some calculation, we can find that the variation of tension force within this elastic film is proportional to the density variation, okay, which corresponds to the wave. Okay. So in fact, the spiral wave we just see reflects the tension force distribution across space. So in other words, the entire bacterial film uses this tension wave to regulate large-scale forces, tension forces. Okay. Yes.
Question you might have mentioned, but is this a 2D system or a 3D system? I mean, I'm wondering, are you getting this buckling in the third dimension, or is it really like a density wave?
So it's quasi 2D, and so the thickness is about 10 cell width. In some sense, it could be regarded as 3D, and the height, if one would like to measure and the resolution is high enough, one might get some height variation. But here we ignore this. We treat it as a 2D system during this calculation. All right. Now to further understand how the waves become synchronized, we resort to the classical Kuromoto model. In Kuromoto model, the Kuromoto model consists of a set of a couple of oscillators, phase oscillators. So here we put these couple oscillators in our system, but add a new ingredient, that is that the oscillators have a non-reciprocal coupling. That is, oscillators are easier to advance their phase rather than delaying their phase. The reason, the biological rationale is that Puller's motor is highly irreversible. The activity is highly irreversible. It's very difficult to reverse the retraction, the extension retraction cycle. So with this assumption, where we put as this step function, okay, this indicates the in non-reciprocal coupling. So here this function is described as by this parameter epsilon. Epsilon belongs to 0 to 1. It measures the level of non-reciprocal coupling. If it takes a value of 1, then that means if the phase of an oscillator would not be affected by any oscillators that's lagging behind. Okay. It will only be affected by the oscillators whose phase is advancing above, on top of it. Okay. So, yeah. The subscripts indicate the position of these oscillators. Okay. So, all right. So this non-reciprocal coupled Kuromoto model can reproduce the tension wave, the spiral wave. Okay. As we can see from this video, it describes the development of a initially homogeneous system from initially scattered wavefronts to the later fully developed, very stationary spirals. So this shows the spiral core space-time trajectory. It indicates the stability of this wave in the simulation. Okay, and the level of non-reciprocity in the model can choose the stability, as we can see that at small values of epsilon, many of the spirals would annihilate rather than keeping a distance away from each other. And the model allows us to further understand how this stability emerges. What we found is that if the distance between two spirals is at an intermediate range, there's a repulsion between these spirals. So this intermediate range repulsion, okay, indicated by these blue triangles. So the range with this intermediate repulsion will increase as you increase the level of non-reciprocity. So we can explain that why we need to have this non-reciprocity in order to have stable spiral waves rather than a fully synchronized system. All right, so that's the story, the second part of the story. Now to summarize, here we have shown two examples how bacteria can make use of the coupling of microscopic mechanical processes to generate large-scale forces and to regulate these large-scale forces. And it could be useful in materials engineering to help design emergent mechanical functionalities. And again, even for microbiology itself, these forms of large-scale forces can be useful in their transport or during their their interaction with the host or the environment. And as flagella, so flagella generates extensile stress, and type 4 pili, as I mentioned, it's pulling around, so it generates contractile stress. So it's very similar to some animal cells that generate contractile stresses. So this can be an analog or a very prototype of the animal tissue to study more general tissue morphological problems. And so finally, I would like to thank my students and collaborators in recent years who have helped us to get here, also the funding sources. With that, I would like to thank you for your attention.
All right, thank you, Yilin. Any questions in the audience? Anton.
Very interesting talk, thank you. In the first part where you showed the rotating sort of patterns, I'm curious, what sort of motility do the bacteria have? Are they swimming through the fluid, through the biofilm? Do they crawl on the surface? Is it a combination of both?
So in the first part,
yeah.
Right? So in that case, the cells are relatively fixed. They do not have relative movement. So what we are imagining is that although we do not have the full picture, microscopic picture, what we imagine is that the cells are those cells embedded in this elastic matrix, they are coupled to a permeating fluid. So the flagella is still rotating, and it can generate forces. The momentum, some of the momentum is transferred to the permeating fluid. Meanwhile, it generates propulsive force to deform the elastic matrix.
I see.
Okay, and that gives rise to the global motion.
I see. So it's a purely mechanical deformation that leads to this motion.
Yes. Yeah.
Thank you.
Do you have a sense of the mesh size of the network?
Yeah. So we at the beginning I show a fluorescent image of the matrix produced by the cells. It's roughly tens of microns of the size.
Yeah, I guess I have the same question, but for the second system. What is your best microscopic version of that? I understand that there the bacteria is like pulling on a string, right?
Yes.
Do you think the attachment point changes? A, and B, do you think it's mostly with the substrate or between them? Or do you have an indication of them?
Yeah, very good point. Again, that's a very good question. But again, I do not have a clear microscopic picture. It's very difficult to image either flagella or puli in this dense environment. Although we can easily image them when they are isolated, but when they are in this environment with these behaviors, we are not able to do that yet. My imagination would be that they are constantly pulling each other rather than pulling on the substrate. Because we did some experiments to measure the attraction with the substrate, and we found that the scale is very small. But I don't know, maybe because the substrate we use is too stiff. We're now trying to study this problem with variable stiffness and see what how or whether the substrate elastic mechanics can feed back to the system.
I was wondering whether you know what's the mechanism that couples the pili, because you introduced this synchronization, and I wonder what's the microscopic mechanism that is responsible for it.
Yeah, that's a very good point. Maybe you know that Puller's motility, it works as a stochastic motor, right? Yeah, many people who study individual cells Puller's activities found that the extension retraction, or all the kinetic quantities, they appear to be stochastic. But that's at a single cell level. It hasn't been documented that Puller's type 4 puli can synchronize in any form like this. So what we think now is that they could be using the mechanosensing pathways to do this synchronization. We have several layers of evidence supporting this Puller's synchronization. They must synchronize. For instance, we use chemical drugs that can trigger instantaneous Puller's retraction, and that can trigger waves in an initially homogeneous system. So this synchronization indeed happens and can result in the waves.
So you're saying the mechanism is that the pili must be mechanosensitive, and then
yes.
they respond to tension, and so that
yes.
synchronizes them.
Yes. Some work, recent work from Gitai and Passat labs, they found that the Puller's motor can be mechanical sensors. Although the full molecular pathway of this mechanosensing is still being investigated, but I guess the cells are using some of these pathways.
Thanks, very interesting.
All right, thank you.
So maybe this is a related question, and I think you sort of alluded to it earlier, but what is this phase that, I know what the Kuromoto model is, I know it gives spiral waves, but presumably it's not the phase of bacterial motors, and you have equal chiralities, those are chiral, so it's just something in a chemical reaction which has an interpretation of a phase that goes from 0 to 2 pi, or, I mean, the Zhabotinsky reaction doesn't have a literal phase. So can you say more about what this phase is?
So here the phases are purely phenomenological, based on the fact that, or the observation that every mass element changes its light intensity periodically. But down to the microscopic level, what's accounting for that? I think that has to do with alert question. That is, it reflects the level of synchronization of Puller's motors in a mass element.
So swarms of fireflies, which also blink,
yes.
and have periodic light intensity, they would be described perhaps by a similar model. Has spiral waves been observed in fireflies?
I don't know. Yeah, maybe not.
I don't know either.
Kuromoto models. So we did an extensive literature search. We do not found any discussion about spiral waves based on simple Kuromoto models. Some versions could produce. Yeah, there is. Yeah, there is a version that involves, that also modifies the way of coupling, that also introduces kind of like asymmetric coupling. But the way of coupling is quite, I would say, unusual. So we can discuss that more.
We should discuss later. But the Kuromoto model has without non-reciprocal couplings, has spiral waves also. Or do you need
without non-reciprocal coupling, we cannot produce spiral wave. It produces global synchronization.
Right. Thank you.
Right. A related question. So, do you think the bacteria is using the spiral core for some purpose?
Yeah, that I don't know. Yeah, it looks very. So the cores, the position are very stable, and if we look very closely at the cores. So we do not see any difference in features, in appearance or density or whatever. So they look very trivial. It's just some spontaneously selected points to be the cause. Yeah.
And I have another question about the first system. So I'm still not clear about what is the synchronization mechanism. Presumably the bacteria are all randomly oriented, and so they are exerting forces like randomly, but how do you then get these collective states?
Yeah, so are you talking about the first, the flagellar system?
Yeah.
Yeah, in the flagellar system, presumably every cell is generating forces at random. So we believe it may involve some synchronization or self-alignment process. For instance, the flagellar bundle direction relative to the cell body could change. It can shuffle, or it can rotate around the body. So if a mass element is picking up some collective speed at some direction, then the flagellar bundles might tend to align with that speed. So that's our physical picture. We don't know whether this is correct or not. No, their cell body is not orienting. So we did extensive measurement. We found that the cell bodies, the cell body orientation are almost constant throughout this motion. There's no relative displacement or relative angular change between two individual cells, two nearby cells. So it's the only thing that can change is the flagellar bundle orientation. That's something we cannot assess or measure yet.
Yeah, sort of related question. So do you only observe the global rotation and translation modes, or is your analysis also sensitive to strain fields inside the material? And are there any kind of correlations in these strains?
So
in the flagellar system.
So the question is about whether one can tune these modes by the strain field?
Not necessarily tuned, but have you observed anything interesting in the strain fields?
In the strain field?
Yeah, in the strain fields. Or is, I don't know what the resolution is, but like whether strains can be measured at all.
Right, that's a good point. Yeah. We try to measure the displacement between two mass elements, and we do not see interesting things in this. In most cases, the displacement is very, very small. So that's why we conclude that they do not have any displacement at all.
Even if you somehow tune the elasticity of the with the polymers or something.
We didn't look into this very carefully. Yeah, maybe that's something we could do.
Okay.
Yeah, thank you for raising this point.
Very nice talk. A small question. You showed some bacteria that are swimming in a droplet. You showed them at the edge of the droplet. Could you please show that movie again? Where it seemed to me that the bacteria are going in two opposite directions, but it seemed that you were saying they are going in one direction. And my question is, do you know what's the function of such a movement at the boundary? What would be the biological function for them doing this type of behavior?
I guess you are,
I think
here, right?
Yes.
Yeah. Yeah, you are right. So here in this video, the cells right at the edge is moving in one direction. It's clockwise. Right? It's like a chain. Sorry. Yes, it's moving clockwise. And the cells in this layer, they are nematically aligned, and they move in two directions. So your question is the biological function
right.
of this.
Is there a biological function, and what's the mechanism by which they are doing this? They are doing this counter-rotating.
So we think that this the biological function of this would be to transport fluid or materials around the colony. So so this video is a small section of this bigger view. So if we take a small section, what we are seeing is here. So if we zoom out, then we will see that the fluid flow is transported around the colony in a counterclockwise manner. Things are being transported in this region. So we think that the flow in this region being counterclockwise rather than clockwise is because the cells, the flagella of cells here, they are pointing into the fluid, and that sheds fluid in a counterclockwise manner. So we think the purpose or the biological function, if there's any, is to transport materials, especially the self-produced vesicles. That's roughly in the size about several hundred nanometers. It's very these vesicles are very slow in diffusion, but with these flows, it can be very rapidly distributed around the colony. Right. That wouldn't require a counter-rotating rings. Yeah.
You're saying it's a byproduct. The bidirectional vortex is from the flagella and the flow. So
yeah, that's right.
There is the Goldstein and Karalush's paper on the bidirectional vortex.
Yeah. Yeah, the flow is a byproduct of the arrangement of the cells in this way. And the arrangement, the self-organization of these cells in this way is due to the coupling with this confinement at this three-phase boundary.
All right, let's thank Yilin again.
Well, thank you.